So, rather than looking at NAMs principally by asking what animal model does this replace, I think it's important to ask, what's the decision category that this improves? So I've grouped sort of 4 key decision areas here. The first is target engagement. Not simply does the molecule bind a receptor or a target, but, do we see meaningful engagement in a human relevant context? So, I've spent a lot of time in the field of antibody discovery and development. And so we know now that antibody-based therapies are getting increasingly targeted, going after very novel epitopes and targets that frankly might not exist in that form in animals. We're also, generating human antibodies. And so, to expect them to go and be a species cross-reactive, they may not also always bind in rodent models, for example. And so people will spend hundreds of thousands of dollars genetically modifying a rodent, for example, to express that specific epitope. And it might take a couple of years to get there, by which time the target is no longer novel. And, you've lost that advantage of your novel target identification. Second is, the functional efficacy. Once you've engaged that target, does it produce that downstream biology changes that we expected? Third is really, we can answer questions about liability identification, I think, much sooner. Are there unwanted biological effects that appear alongside the efficacy? I think that's becoming increasingly important as we develop more complex things like ADCs and bispecifics and T-cell engagers, where, relevance, the species relevance can become increasingly challenging and liabilities, increasingly harder to predict. And finally, I think, exposure, tissue interaction, does your drug actually get into the relevant compartment? Can it penetrate tissue? Can it pass through blood vessels and get into the surrounding tissue? These are real 4 key areas where I think NAMs can materially change and improve development decisions. But there's an important qualification to all of this. I think more complexity is not, automatically better for a model. If you want to understand simple target binding, then a simple biochemical assay might be, perfectly fine. Or a cytotoxicity question, just putting your target or your therapy into a 2D culture might also be, perfectly sufficient. But once you get into things like immune trafficking, vascular toxicity, questions where, spatial biology becomes more important, you have to choose the right model for the right question. So I don't think the future is one where we simply kind of make models more complex. I think it's ones where, we start to appreciate that so the least complex model that can reliably answer a question that we actually need answered is the one that we need to use. And that's really what fit for purpose should mean. So, and this brings us to validation, which Mike also spoke a little bit about. And that's really the heart of today's discussion. I think there's lots of scientifically interesting and beautiful models out there. But being interesting and cool is not, necessarily what's needed when it comes to the appropriate tool for drug development. It's really first biological relevance. Does this model have the components that we need to answer the question? Can we characterize it reliably and reproducibly? And are we able to benchmark it against known human biology and really have that clear context of use? And then, what is the actual decision that we're trying to qualify from this and inform this model for? And then finally, and I think most importantly, is adoption. So can my model actually be used in another laboratory? Can they reproduce the same results? Does it easily fit into their workflows? Is the cost benefit analysis and the economics of using that model make sense?